Dr. Ed Rogers is a research fellow at the Institute for Life Sciences working on super resolution in microscopy. Ed, what is super resolution?
In this context, it's looking at things that are below the wavelength of light. So when scientists normally look at biological cells or things like that, they're limited by the wavelength of the light that they're using. They can't see things smaller than a certain size. And what we're trying to do is break that limit, allow them to see the functional bits of cells that they can't otherwise see, basically. So watch how cells work on a scale that's previously been invisible.
What impact is this having on imaging?
So this is a very hot field at the moment. Lots of people want to do this because you can see the dynamics of cells, so you can see how a brain cell signals to its neighboring brain cells. People knew they did it before, and it's something to do with transfer of neurotransmitters, but what is the actual mechanism of transfer from one neuron to another just is not visible with conventional techniques. And this is the sort of thing that we can see with these super resolution techniques.
So how are you going about this research?
So my work is very much on the optics side of microscopy. So what we're doing is we're taking a traditional microscope, which you normally shine a very normal beam of light into it, a flat beam of light, and we're sculpting the light that goes into the microscope, so that instead of forming a spot with the standard limited size, we can form a spot that's much smaller. And then if we scan that spot across a sample, we can resolve, we can see things that are much smaller, and basically the size of things we can see is the size of the spot that we can create. And if we can make the focal spot smaller, we can see things that are much smaller. And we can do this without intruding in the cell, and without putting too much stuff into the cell that changes its function.
So what's the significance of all of this?
This will allow scientists, so in the biological sphere, this will allow scientists to understand processes that they can only guess at now, and actually see them working, see it, test their theories, if you like. But this has applications outside of biology as well. These same technologies can be applied in nanomaterials or even nanomanufacturing systems for manufacturing nanoscale structures with light.
Ed, thank you very much.
Thank you. Thank you.